Hi everyone. In this video, we will discuss about the two principal mechanisms of the adaptive immunity against the intracellular bacteria. So these two mechanisms are the helper T cell and also the cytotoxic T cell. When the bacteria go into our body, our phagocyte will normally recognize them and engulf them. So after the phagocytosis, by right, normally these bacteria will be captured in the phagosome, and later on, the phagocyte is going to produce the lysosome to fuse together with the phagosome and digest this bacteria. So this is what normally should happen, and the bacteria should get killed inside the phagosome. However, there are some of the bacteria that can actually evade or escape from the digestion of this lysosome. So these bacteria will be able to stay inside the phagosome. So these bacteria will stay inside the phagocyte, in other words. So at this point, we can say that these bacteria is actually the so-called intracellular bacteria. So to deal with these intracellular bacteria that actually stay inside the phagosome, we will need the first mechanism, which is the helper T cell. So when the phagocyte is having the bacteria inside their phagosome, then they are going to extract the antigen. So they will present this bacterial antigen on their surface through the MHC class II molecule. So this MHC class II molecule and the antigen will be recognized by the T cell receptor of the helper T cell. The helper T cell is going to use their T cell receptor to recognize the antigen that is being presented on the MHC class II molecule of the phagocyte. Then at the same time, the CD4 marker of the helper T cell will bind to the MHC class II molecule. So this is the double confirmation. So after the double confirmation, this helper T cell will release the interferon gamma. And this interferon gamma is essential because it is going to activate this phagocyte in order to destroy the bacteria inside the phagosome. So what the phagocyte will do is that they will produce the nitric oxide, they will produce the lysosomal enzyme, they will produce the ROS in order to kill these bacteria, these intracellular bacteria inside the phagosome. So by doing this, our T helper cell will be able to help to eliminate these intracellular bacteria. However, there are also some bacteria that can still manage to escape from this. So when these intracellular bacteria escape from the phagosome and go into the cytoplasm, then now this intracellular bacteria will be staying inside the cytoplasm of the phagocyte. They have actually escaped from this phagosome. And to deal with this, intracellular bacteria that actually escape into the cytoplasm, we cannot rely on the helper T cell. We will now need the second mechanism, which is the cytotoxic T cell. When these bacteria escape into the cytoplasm, it will cause this phagocyte to present their antigen on the MHC class I molecule. So those cytotoxic T cell that is already activated will recognize this antigen that's being presented on the MHC class I through their T cell receptor. Once they recognize the antigen, then at the same time, they will use the CD8 marker to double check. So the CD8 marker will bind to the MHC class I molecule, and with this double confirmation, the Cytotoxic T cell will start to release the perforin and also the granzyme. The perforin is going to create the pores on the surface, on the membrane of this phagocyte, while the granzyme is going to get into the phagocyte itself and induce the apoptosis. So that means that these phagocytes, together with the intracellular bacteria, will be eliminated. The whole cell will undergo apoptosis, and the remnant of this phagocyte and also the intracellular bacteria will later on be engulfed by other macrophage or other phagocyte. So with that, the helper T cell and also the cytotoxic T cell will eliminate those intracellular bacteria.